Good morning, viewer. Welcome to our active English version. Here's a really amazing news and warning news for our viewer. Researchers at the DTU National Food Institute have developed a faster way to identify bacteria that can both support fermentation and boost vitamin B2 level in sour drinks. Yeah, viewers, in this study, they analyzed the microbiome, the complete bacterial community from Bumblebee by isolating individual microbes in microscopic droplets and testing their ability to produce the vitamin. Our research shows that it is possible to screen entire microbial communities directly and rapidly, and that promising bacteria can be identified from environmental samples without prior isolation and analysis of individual bacteria. So this can make the development of new starter cultures faster and more targeted, says Associate Professor Klaus Hanier Bang from the DTU National Food Institute. The findings were published in the journal LWT Food Science and Technology. Yeah, researchers have discovered promising bacteria in bumblebees. Plant-based dairy alternatives often contain fewer vitamins and minerals than cow's milk, with vitamin B2, that is called the riboflavin, commonly lacking. To address this, the team focused on finding bacteria that could grow in sour drinks while naturally producing vitamin B2 during fermentation. So they turned to bumblebee gut bacteria as a source of candidates. Amazing, sir. Yeah, bumblebees. Bumblebees live close to plants and their guts contain many microorganisms that are already adapted to plant-based environment. That is why it was interesting for us to test whether we could find bacteria in bumblebees capable of producing vitamin B2 in sardines, says postdoc Hang Zhao from the DTU National Food Institute. To carry out the screening, the researchers adapted a technique known as droplet screening. Each bacterium from the bee gut was enclosed in a teeny droplet that acted as its own miniature culture chamber, allowing millions of cells to be tested within hours using a microfluidic system. Unlike conventional agar plate-based methods for microbial cultivations and screening, we encapsulated the bee gut bacteria in microscopic droplets so that each droplet contained only one bacterium and acted as an enclosed culture chamber. In this way, the individual bacterium could be analyzed at ultra high speed by using our microfluidic screening platform, enabling us to screen millions of bacterial cells within the just few hours, says Hang Zhao. Bodown said, when this exclusive report, FTV English, Delhi, Bharat.